Hello, everybody. So I just wanted to create uh, just a quick revision of the first lecture in microbiology. And so I'm going to start. This is the book that she recommended for our semester. She mentioned also that we have to answer those questions at the end of the presentation. And so she probably have gone over most of these questions. So she began by saying most bacteria they have different shapes, so coccus, rod, bacillus, and spiral, and so these stand for different shapes of the bacteria and how they live. This presentation, she said that it's corresponding to chapters 12 to 16. So first thing she dived into, which is the first shape of how a bacteria can exist, the cocci. So spherical or oval, and you see that the different pictures show different arrangements, and so different bacteria that we're going to study in the future are going to be aligned in different arrangements. So the first picture here shows how it divides into two pairs, or it can be, let's say, creates a long strain. And so different bacteria can take those different shapes if they're under this category of spherical or oval bacteria. Okay. And so a bacteria is a prokaryotic cell. It's unicellular, microscopic. It produces by binary fission, which is splits in half, basically. Simple structure. It has a nuclear body called the nucleoid. It's not bound by a nuclear membrane. So what this means is that it's surrounded by histone proteins, and so helps pack this nucleoid, this piece of DNA that is free to move around. It's inside the structure, and so it's held by histone proteins. And I think you've taken this back in the biology when we talk about nucleosomes, histones that are H1, H2, H3, H4. So these can be used to pack this DNA. She also mentioned that all prokaryotic cells are haploid, which means that they are not diploid. They have one N, not two N. And so there's no nucleolus in the bacteria. The next thing is, she said, is a typical bacterium consists like a cytoplasmic membrane, has a peptoglycan, which is a complex that surrounds this bacterium cell. And so it basically forms a structure around the cell cytoplasmic membrane. Okay. And so this peptidoglycan can only be the outer membrane, or in some other forms, such as gram-negative bacteria, there's an extra membrane covering that peptidoglycan layer. Okay, so a fluid cytoplasm containing a nuclear region, nucleoid, and numerous of ribosomes. So ribosomes are very necessary and exist inside a bacteria to help produce a lot of proteins. Some external structures that are found, glycocalyx, capsule, flagella, and pili. She said that glycocalyx is not the same as the capsule. Glycocalyx is thinner than the capsule, and the capsule is a bit thicker. And so she also said something about the capsule, that there are antigens, and so the capsule is very thick, and because of the capsule being so thick, those antigens are not easily captured by immune cells of the body. And so what happens is that the immune cells of the body cannot fight this bacteria and helps it to stabilize inside our body and cause effects of diseases inside of it. Flagella causes movement. Pili helps in attachments. The cytoplasmic membrane, this is just a structure that basically surrounds this cytoplasm of the bacteria just directly. It is a fluid, phospholipid bilayer, and has proteins. It does not have sterols, except for a bacteria called Mycoplasmas. The electron transport system is located in the cytoplasmic membrane. And so electrotransport is used for basically producing of ATP. So this is basically the structure of the bacteria she showed us. And so if you see that the nucleoid exists inside of this bacterium, is basically here in blue, all of the dots that you see in dark are the ribosomes used to produce the proteins. And so the first thing that we face outside the cytoplasm is the cytoplasmic membrane that we talked about. After that comes a layer of the cell wall. And so the cell wall can be made up of peptidoglycan and then maybe a capsule at the external level. And then you have all of those hair-like structures called pili, which help in attachment of the bacterium to some structures. For example, as she said, when the bacterium is not able to be pushed out of the body because of certain fluid flush, such as saliva or urine, pili is the main reason, because pili helps it attach to the structures. And so that's what's the function of the pili here. Flagella helps in movement. The bacteria cell wall basically made up of peptidoglycan, which is a complex. What else? Yes, so the two strains of bacteria that lack a cell wall are the Mycoplasmas and Urea plasmas, okay? What else? Let's go. Peptidoglycan, what is it? It's basically a complex made up of 6 shaped rings. And so, no, 6 shaped rings. They're, I think, hexagons, okay? And so these hexagons are NAG molecules and NAM molecules attached to each other, one after the other consecutively, to form a very thick layer, okay? And I'll show you. For example, these are the peptidoglycan monomers, NAM and NAG, and they are attached to each other to form this huge, thick peptidoglycan layer on gram-positive and a thin peptidoglycan layer on a gram-negative. So gram-positive bacteria. Okay, so here it says that they retain the dye and appear purple. This is because if you dye a gram-positive bacteria with a violet dye, and so then try to wash it out, what happens is you can't wash that violet stain out because of this very thick gram-positive peptidoglycan layer. And so they will stain purple. So this is the reason. So you have the structure of the gram-positive bacteria is that it has a cytoplasmic membrane around the cytoplasm. The gram-positive bacteria cell wall is very thick. Why? Is because the peptidoglycan molecule is very thick. And so just remember that positive means more, so more peptidoglycan. And so in the peptidoglycan molecule, we see that there is very important instructions that we have to pay attention to. Antigen molecules. The antigen molecules in gram-positive bacteria are the teichoic and lipoteichoic acids. And so these are very important because they initiate response in our body, such as inflammation and fever. And so some other structures in the body are the surface proteins, okay? And so they're very useful. Okay, so that's the structure of the gram-positive bacteria. And then the gram-negative bacteria, on the other hand, they decolorize during the gram staining procedure and appear red. So what happens is we do the procedure, and I'll show you guys in a bit what she said about the experiment that we're going to take, how to figure out what bacteria is gram-negative and what bacteria is gram-positive. Is you carry out this colorization or dyeing process. And so gram-negative bacteria cannot retain that violet color because of the thin peptidoglycan layer. And so it will stain red when we are colorizing the 2 strains of bacteria by red dye. So the red cannot affect the gram-positive bacteria because there's a lot of violet, so that you can't change that color into red. But then the gram-negative bacteria is washed out of the violet. So as soon as you put out red dye on it, it takes on that color and becomes red. So that's why it stains, and that's how it's shaped on the slides. So gram-negative bacteria cell wall structure is made up basically of the cytoplasmic membrane first, then comes the thin layer of peptidoglycan, then comes a protoplasmic, I think, space, which is here, just between the outer layer and the peptidoglycan, which is filled with enzymes that are used to probably, I forgot what the structure, what the function of these enzymes here. I think something to do with antigens or something like that, but I don't remember. But there's the cytoplasmic membrane, the peptidoglycan, the protoplasmic space, and then comes this outer membrane. This outer membrane is made up of so many important structures such as proteins, and the antigens here are the lipopolysaccharide, and so this one is also very necessary for the function of the bacteria. If it's released inside the bloodstream of the person, it can cause death. Okay, and same with the other antigens, can cause inflammation and fever. So this is what I'm talking about. So if we are trying to differentiate between the two bacteria, so what we do is do this procedure. First thing, we expose both of them to crystal violet. They both take the color. Then we treat them with iodine. I don't know what iodine does, but forgot what iodine does. I think, oh yeah, so yeah, iodine would wash off that crystal violet as much as it can. So if it's very thick, the iodine cannot take off the crystal violet in the gram-positive, so it will stick into the thick peptidoglycan layer. Unlike the gram-negative, it's very thin, so it will wash out. Now we counter stain. We're using red, so because of the gram-positive cannot release the crystal violet due to the very thick peptidoglycan layer, it will continue to stain this color, but then because the crystal violet was washed out from the gram-negative, it stains red. So this is how it appears on the microscope. So you can see that the gram-negative is red and the gram-positive is blue. And so that's how we differentiate between the two and see that and know that which one has the thicker peptidoglycan layer, hence which one is gram-positive and which one is gram-negative. Gram-positive bacteria can be divided into these groups, and we're going to dive into them across the semester. I forgot to take a picture of the other slide for gram-negatives, but then these ones here is a third slide after. These are not classified into whether it's gram-negative, gram-positive, and so they're just categorized here. Okay, so these two bacteriums that we've gone over today are called the Clostridium and the Bacillus. They both are very necessary for the release of endospores or spores into the environment, where the environment is very harsh for the existence of those bacteria. So they produce the spores because those spores can take on a very harsh environment, and so for the survival of those bacteriums, they would produce those spores so that they can exist in this environment and not be killed. So what happens is the spores are produced inside, and I'll show you guys how this happens. So it will get involved, thickening of the spores inside of the bacterium happens until it becomes very thick. Then the outer surface of the bacterium just moves away and dies off to leave off a spore to be in the environment until it's very safe for the spore to germinate and produce the same bacteria again. So this is how it happens. And then the endosporium would be released when this outer structure gets exposed to the harmful stuff, can't resist, breaks down, and then this is released in the environment. It can be dormant for long periods of time. Structures on the cell wall, yes, that's what I was talking about. Glycocalyx, this is slime layer in those strains of bacteria, and it's thin compared to the capsule. It's a viscous, thick covering surrounding a cell composed of polysaccharides. So these are the structures on the cell wall, which is the most external part. Structures outside this cell wall: flagella and pili. Flagella for movement, pili for attachment. Flagellar attachments, they can be of different types, and so they're just named into different categories according to what is present on the external part. Pili here, they're found on all gram-negative, absent in many gram-positive. Remember that. The pilus has a shaft composed of a protein called pilin. So pilin makes up the pili. Adhesive structure, which helps in the attachment of the bacterium to different surfaces. The pili can be of different forms, either for attachments or for conjugation. So pilis are actually used for sexual activity of the bacteria. So pilis are used for basically having sex between the bacteriums. These are very dangerous, though, sometimes, because through this pilus or conjugation pili, the genetic material can be transferred from one bacterium to another. Effects of oxygen on bacteria, they are categorized into how they are able to use the oxygen. So obligate aerobic bacteria, they cannot survive unless oxygen is present. Obligate anaerobic bacteria, they cannot survive if oxygen is present. So they need no oxygen to survive. So absence of oxygen is where they thrive. Microaerophilic bacteria, they require lower concentration of oxygen. Here, facultative anaerobic bacteria, they can grow without oxygen, but also can use it if it's present. And aerotolerant, these bacteria cannot use oxygen for the growth, but they're not harmed by it. So oxygen will be present, they don't need it, it exists, and they can grow. So bacteria is also categorized into nutrition. In this semester, we're not going to be talking about autotrophic, we're going to be talking about heterotrophic. And so this is basically it for this lecture. You need to know shapes, you need to know gram positive, gram negative, how to differentiate between them. And the gram reaction is related to the structure of the cell envelope. Basically, there's peptidoglycan. There's a lot of peptidoglycan. There's gram positive, less gram peptidoglycan, gram negative. So pathogens of infection. So here what happens is bacteria can be adhesive. They also are invasive. They invade and spread in tissues. Immune system invasion, which means evasion, which means that they can survive inside macrophages. Toxin enzyme production, they can produce toxins. Multiplication at sites of infection, they can multiply and form biofilms. Stimulation of immune response, which means that due to their antigens, immune cells in our bodies can trigger an immune response within our body because of their presence. So for the next lecture, they said that we have to read chapters 18, 19, and 20 of the Murray textbook. So I hope this was useful for all of you. I hope I can continue to do those because these are very useful for us, I think. So